So far in this module, we've talked about different characteristics of DNA and different types of genomes. And for the next part of this module, starting in this video, we're going to dive a little bit deeper and talk about different techniques or methods that are used in molecular ecology studies. And we'll start with the techniques of PCR and Sanger sequencing in this video. So PCR, or it's just short for polymerase chain reaction, what it does is amplifies one specific region of the genome. So you make a lot of copies of that one particular region. You can then use those copies to do other things, do data analysis, things like that. So on the right, what I'm showing is a schematic of what happens during PCR. I'm pretty sure that all of you have had some kind of exposure to PCR before in your previous classes, but we'll just do a quick refresher here. So let's color code this. So our original template DNA we'll do in blue. So that's shown here at the top of the image. We have our original DNA sequence. That's often called your template. We have these short sequences called primers. So you always have a forward primer and a reverse primer. And those primers are what is specifically designed to target that specific region of the genome that you wanted to investigate. So your primers are these nucleotide sequences that are specifically designed to bind to that template DNA specifically on the region of the gene that you're interested in. Another component of this process, once you have those primers bind, what you end up making are new DNA sequences. So these regions here are the new DNA that has been sequenced off of our template based on whatever particular region the primers bound to. So basically, if your primer binding here, it's going to extend the DNA that way and extend it that way until you end up with these long new stretches, the yellow new template, new synthesized DNA sequences. And then what happens is this happens over and over again, and that's how you end up with so many copies at the end of this newly synthesized DNA. Those new copies are then what you, before we get to that point, the new copies are made in this machine called a thermal cycler. Okay. And what you do when you make those new copies, you run that PCR in the thermal cycler. What you want to do next is run out the results of your PCR, so that reaction that you made, run it out on a gel to check, first of all, if you have any PCR product at all, and then also to make sure that the product is the right size that you expect. So in this science Drake meme shown here, on the right is an example gel you might run from your PCR. On the left here, that's called your ladder. So there are basically bands of specific lengths in that you're loading into the gel that you can use as markers to see how long your, at this point, unknown PCR product is. So first, like I said, you want to check and make sure you have any PCR product at all to ensure that your reaction actually worked. So if you see nothing on the gel like this, except for this faint band at the bottom, that's called a primer dimer. That's just the primers binding to each other, but you haven't actually created any new DNA. If you have nothing on your gel, that's bad or sad, right? It didn't work, we would have to redo it, try it again. If you do end up with something like this, where you have a nice bright band, the stronger or brighter the band is, that means the more PCR product you actually produce in that reaction. If you have that band, and when you compare it to these ladder sequences, it's the right length or size that you think it should be based on the primers that you designed, then we're good to go. We can move on to the next step. So just to show in a different way what it looks like when you run your PCR out on a gel. Remember, DNA has a negative charged because of its phosphate group. And so because of that, DNA runs toward this positive electrode here. So you always load your samples up here in these wells, and then they run this way. Here on the left, we still have our size markers or our ladder. They're these specific lengths. So that one right there is 400 base pairs long. And then you can load your samples in these other wells and see if the band that shows up is the right length. So for lane 2, for instance, if we had designed primers to amplify a region that was 400 base pairs long, and here we're seeing on our gel that that PCR product is 400 base pairs long, then that's what we would expect. That's good, and so once we checked, we can then move on to the next step, which would be Sanger sequencing. So the PCR is usually not the only thing we want to do. Usually that PCR is a middle step to help us get to our end goal, which is normally to figure out what the actual DNA sequence of that particular region we amplified is. And so here, this is an example of something called a chromatogram. And this is what actual Sanger sequencing data looks like. Essentially, there are 4 different colors that you can see here. There's red, green, black, blue. And each of those colors represents a different nucleotide. So we have T is shown by red here, the green is A. And so what these chromatogram data look like are you have these different peaks. Each peak corresponds to a different nucleotide, and then the color of that peak tells us what nucleotide was present there. So if we look here under the A region, the first 2 peaks are red followed by a black. That means that first 3 nucleotides there are TTG. So that's how you read this chromatogram data, and that's the data that you get as a result of Sanger sequencing. And remember, that DNA sequence you get is only for that particular region you amplified based on the design of your PCR primers. So in this example, if we want to give it some more context so we can think about what that might look like more in the real world, say we have the state of California. Yes, that's my excellent drawing of California. And say that these 2 sequences, A and B, were taken from 2 different individuals, and say that this A was taken from a snail from Northern California, and B here is taken from a snail in Southern California. Okay, so what we can do then is once we've done PCR, so we're looking at the same DNA region for both of these individuals, but what we can see here indicated by this black arrow is that some of the DNA in those individuals is different. If you think back to earlier in the module, we talked about there can be nucleotide substitutions in the DNA. That's one of the things we're often looking for when we're looking at this Sanger sequencing data, is to see if there are any nucleotide differences among individuals that are the same species, for instance, but they're from different geographic sites or different populations. And so in this example here, we see that this same nucleotide site, we have a C nucleotide for the Northern California snail and a G nucleotide for the Southern California snail. And so if we did this a bunch of times and got a bunch of samples from Northern California, a bunch of samples from Southern California, and saw that they all had this same nucleotide difference where all the Northern California ones had C, all the Southern California ones had G, that would tell us that this nucleotide difference is potentially playing a role in how these organisms from these different geographic sites have evolved differently. And so we'll talk a lot more about that later, but that's the one example of what Sanger sequencing can give you. It can identify these different nucleotide differences among your samples.